Once I was told by a professor that I have too much lateral thinking for my own benefit. Now I think it's the opposite. I think students require to have it, because if you cannot think in 5 different ways at the same time, you're not going to solve it.
Today's guest is someone whose work sits at the heart of the most profound questions in modern neuroscience. How does stress shape who we become, and can trauma echo across generations? Dr. Nikos Daskalakis is a psychiatrist and molecular neuroscientist at Harvard Medical School with joint appointments at the Broad, MIT, and the National Centers for PTSD. His research connects genetics, epigenetics, and neurobiology to understand why some people develop post-traumatic stress disorder, while others exposed to the same events remain resilient. Over the past 2 decades, Nikos has helped define the science of stress, from early animal studies exploring how life experience programs the brain, to large-scale human analyses that link molecular signatures in the blood to what's happening deep inside the brain. His 3-hit model of vulnerability and resilience reframes how we think about trauma, showing that our biology, our early environment, and the events we face later in life are constantly interacting to shape mental health outcomes. In this episode, we explore how PTSD differs from depression at the cellular level, what glucocorticoid signaling reveals about the biology of stress, and how epigenetic inheritance might transmit the marks of trauma from one generation to the next. It's a conversation about science, but also about humanity, what stress can teach us about adaptation, survival, and healing. Welcome to Neural Compass. I'm Mark Jacobstein, founding president of Jimini Health. Let's get into it. Tell me about how you ended up looking at what you're looking at and spending so much time thinking about PTSD and some of the genomic drivers for the challenges with PTSD.
Yeah. So basically, very nice to meet you through this media, and also, I really like the podcast and your work. So my journey, all my career, more than 20 years, I have been always working on stress, and this kind of serendipitous happened that I was in med school, and I saw an advertisement for an exchange program called Erasmus between our medical school in Athens in Greece, and in this particular case, it was with Holland in Leiden University. And then, I applied for it to see if I could fit. And the professor of neurobiology told me, I'm not sure if this is going to be a good fit for a med student because you guys don't do research and, you are good with books, but, but by coincidence, I have done some kind of a summer program in the island of Crete that I kind of like my family's from. And I'm like, oh, but I have done this. Maybe I qualify. Who knows? And she's like, okay, I'm going to give you a try. She was like a very good scientist. And she thought like, oh, we can not send to Holland any random person. It needs to prove that they're good. And I'm like, oh, yeah, give it to me. And, all this in Greece is like free work, volunteering for it. And it was on early life stress. And it was mouse models of early life stress, understanding like how to handle rodents. Rodents, in this particular case, was rats. And, was fascinated about their behavior, all sorts of things. And, it was my natural calling, let's say, as a topic. The brain, neuroscience, everything. And then I got the opportunity to go to Holland, and then I ended up being in this exchange program to the professor Ron de Kloot lab that he's kind of like one of the, the major figures in stress biology, that he ended up my advisor, my PhD advisor. So I go there and I see how the North Europeans do research. And I also got this idea that is not so much in the curriculum in Greece, that, a med student can do their own trajectory. Like you can define your own trajectory. North Europeans, especially in Holland, they have that mentality. In Greece, it's more like, oh, you have to be, strictly speaking, a doctor that you can be relied on. So everybody has to. It is explained to us in Greece that this is because Germans created the Greek universities, because we had a German king, but I don't know if that's the reason. But I really like the Dutch way, like, oh, I can set up my own project. I can pick out my own courses. And, since then, I kind of like applied this principle. And then, I did this project. It was again on early life stress, but it was more about the neonate brain, not about the adult behavior. It's like when early life stress happens, what exactly happens in the brain the moment that this early stressor, maybe the first stressor that an organism experiences, how does it play out in the brain? That was the
It's fascinating because one of the papers of yours that I looked at was this 3-hit model that you proposed, and very loosely, the 3-hit model was in order for somebody to end up with sort of adverse reactions to something like PTSD or other types of stress. You have genetic predisposition, you have sort of early life encounters with stress that essentially probably tweak what's happening already from a predisposition perspective, and then the sort of proximal cause. And it's funny, I was reading that paper and I was struck. I can't believe I remember this, but in 1989, I took a class with a psychology professor named Jerome Kagan. So he's a Harvard undergrad. And honestly, the only thing I remember from that entire class was this study that he talked about where cortisol levels measured in newborns, human newborns, were extremely accurate for predicting levels of anxiety later in life. So if you're an anxious 3-year-old, as measured in a very objective way by like your cortisol levels, your chances of being anxious when you're 5 and when you're 15 and when you're 30 were also much, 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 much higher. And there's a lot of talk in the course about sort of nature and nurture. Anyways, I'd love it. So tell me about the sort of 3-hit model and what it was that you learned. Yeah.
So what you learned there was like the idea of programming, right? That basically, while you are encounter the first stressor, that first stressor encounters your biology that you were born with, some genetics and some epigenetics that your brain cells have. That initial response sets a set of changes that could predict what would be your future encounters, because they are trying to program an individual. Say, okay, you were born with these genetics. Here is your environment telling you what you're going to have. So let's program this, because we have to predict for you to be prepared to later.
Yeah.
You have those studies like more typical cases in diabetes, right? That an organism that is the fetal environment or, limited resources, either directly or through the mother while there is the fetus, gets the signal, very low nutrients. That organism is getting organized and programmed to, in the future, whenever there are nutrients, to kind of absorb them to the highest degree. But if this individual ends up later in life encounter Western diet, diabetes is born, right?
Right, and that's, I think it's the Dutch hunger study, right? Was the famous one that is where this was first established, and this sort of I almost think of it as like Lamarckian evolution, like the mother father, I guess in this case specifically the mother, is passing on genetically, although I guess epigenetically, the nature of the environment to the child in a way that we don't think of as traditional, like Darwinian evolution, DNA exclusively. Talk about that briefly, because we're also going to talk about some other really fascinating Holocaust studies that you did on sort of epigenetic sort of transference from generation.
I want to add one thing, is that what we're discussing here is more like what was called like the diathesis stress model. Basically that, in this situation, the adverse exposure will predict only one outcome. So like either you would be as normal, or you will be not prepared to the environment you will encounter later. But the twist that was happening while I was doing my PhD studies, eventually, again in Holland, was like I was saying, oh, that's very deterministic. And like to write my thesis and kind of like propose what I'm going to do, I was reading a lot of developmental psychology books. And there was this Jay Belsky, and he's a professor. I don't know where he's now, and there was also Joyce and Ellis, similar thoughts, but they, found it fascinating. One was calling it there is not only good and bad genes, and the others were calling that kids are either dandelions or orchids. that kind of fascinated me because it gave this possibility that more outcomes are possible.
Right.
Meaning that if you have, let's say, some genetic background, to be a dandelion, the early exposures, the late exposures don't matter. The range of what environment can do in that genetic framework is limited. But if you're an orchid, you can be in a situation not only like the Athens stress model say that there is this determinism that you will get that early hit and then orchid is ruined, right? No, there is the possibility that actually you can encounter a certain combination of environments that will match or mismatch with what you'll encounter later, and that will eventually have all the possibilities. You'll have the possibility that you can be Van Gogh, right? Or you can have bipolar disorder, or both, right? And also another concept that I explored in that paper, and this it's one of my most well-cited papers. I wrote it in my youngest age. Is that for every outcome, the combinations are different. Like for your creativity, a different 3-hit play out has happened. For your arithmetic skills, for your metabolic profile, that every outcome that I tried in the lab, there was a different story that could explain it, right? And
It's
hence I kind of like
fascinating to see too, Gary, because this paper was from 2014-ish, if I'm remembering correctly.
2013, yeah, 2013, yeah.
To of course the ability to do high throughput multi-omic work now has changed, and we can look at sort of epigenetics at the same time that we're looking at genetics and protein expression. You can do single cell stuff. It'd be really, and of course with AI advances in AI, we can start to look at sort of polygenic interactions, and I suppose people are probably looking at polygenic methylation interactions.
Yeah.
What a fascinating time to start sort of pull a lot of this apart.
Back then, I didn't know very much math. I was like very, a medical doctor, knowing my statistics. I could do my experiments with my own hands and stuff like that, and have a lot of theoretical thinking. But when I came to the US, they told me like, you have to make a choice. NIH here is, what they told me 10 years ago. Now we can change our discussion there. They told me, stress research in the US, it has to be medically relevant. PTSD is your topic. Like, don't go in schizophrenia, don't go in autism, go to PTSD. There is where you should go. And I was like, oh, PTSD, I guess, makes sense. So then I applied for labs, and I was placed in Mount Sinai in the lab of Rachel Yehuda, that she also is also famous for her work in the Holocaust survivors and also their offspring in relation to the biology, mostly. She told me like, oh yeah, cool, you are coming from Ron DeCluse's lab, but we are looking for a bioinformatician. What about your skills in computers? And I'm like, oh, I actually haven't done anything, but I'm willing to try. And she gave me the chance, and then I started getting a lot of PTSD data from blood. And then I'm like, as a neuroscientist, I was like, oh, what blood can tell us for the brain? And Rachel was telling me like, oh, but this is what we have. There is no more. But I was also collaborating with researchers that animal researchers don't get blood samples, but we had the collaboration that, we said like,
animal researchers can do full dissections and have access to the tissue. It's actually really interesting. A couple things I want to touch on here. One is that because in, of course, human research with live human beings, you aren't going to go take a brain sample. It forces you to look at the blood and see if there is signal. And of course, I spent a lot of time at Guardant Health, which was working with circulating tumor DNA in the blood. And it turns out there's so much more signal for what's happening in all of our other systems in our blood than we ever realized. You never, pre-modern machine learning, you never could have pulled these signals out. But now you can, especially when crossed with sort of, it's like inexpensive omics plus inexpensive and astonishing ML. And it turns out there's these faint signals to be seen in the blood in all sorts of different interesting ways. I love that happened. But the other thing I want to...
But the combination of what I was in front, there was my science paper 10 years later. But that particular moment, I was like, okay, I guess the blood-brain stuff, I'm going to do it with an animal model. And then I will do my serial analysis of the blood. But I couldn't predict that in the meantime, I would learn that machine learning can do transcriptomic imputation, and you can predict things from the blood, from the genetics. Eventually, when I was doing this, there were no post-mortem brain studies, right? So then I was like, oh, that's a critical thing, but maybe machine learning can fix this gap. At least maybe we don't have samples, but maybe we can predict them. But then as I was developing the lab, whatever was missing, I was like, oh, we have to have this. And then, 10 years later, I did the study that, ideally I would want to do it like 10 years ago, but you have to be patient.
Yeah, no, it's amazing. And actually, I maybe just for the folks who are listening to this, if you can explain transcriptomic imputation, because it's incredibly powerful when you think about the limitations that we have in terms of accessing tissues in living human human samples, et cetera. But the ability to use this for your work.
Yeah, so imputation is an ambitious concept. I think when I say to mathematicians, they say, are you sure that you want to use the term imputation? But, the way we use it in genetics is like we rely on a certain relationship that the genomic data have and utilize it to increase the number of variables that we... So, for example, a typical example in genetics is, everybody knows the LD structure and the genetic correlations, meaning the correlations that SNPs have, because obviously they are inherited in blocks. And SNP to SNP imputation relies on, in one data set where we paid all the money, we have everything measured, but in modern times, normally we just measure these 500,000 SNPs. Can we predict the others? Because obviously we care about all the possible associations.
Right. I mean, I think of it as a form of, basically, it's extrapolation, right? And where the lossiness of the extrapolation it turns out is less than you might guess in a lot of these biological systems. Anyways, I think it's probably a longer conversation, but a fascinating topic for where your field can go when you're trying to understand what's happening on a cellular level in human beings where you can't access all the tissues all the time.
Yeah, and also when you go SNP to SNP, you're like, okay, I was inside this modality, so I stay inside this modality. It's exciting, but not as exciting when you can predict another modality. Because you realize in foundation relationship, for example, that genetics have to gene expression is written in the code.
Yeah.
So then basically you say, okay, I have the code. There are reference data sets that talk about this tissue, that tissue, that tissue, that tissue. So you can just use the code to predict what would be the actual value. So you go from a 0, 1, 2 variable to a continuous variable of expression. There is not really like measured expression, but it gets you somewhere, right? It gets you to a tissue, it gets you to a cell type, it gets you to where the biologists want to be, like understanding what's going on. It gets you to which developmental period, what tissue, what cell type. And then you have to do the experiment because guess what? When you are eventually doing the experiment, you realize that, okay, transcriptomic imputation was right to some degree, but life happens. So these people will not be exactly as their genetic code predicts. They will have things that relate to stressors, medication, symptomatology, comorbidity with other diseases, comorbidity of diseases of the brain and the periphery, and all those exciting
it gets incredibly complex to pull out all the confounders. It's one of the joys of biological research. So maybe just to pull this back then, another thing from the earlier conversation I think is wonderful is that you chose PTSD. I mean, first of all, it's an incredibly important problem. I know, obviously, you've done a lot of work with, say, folks from the military where there's a lot of challenges with PTSD and very important work. Modern life leads to lots of trauma. So there's many, many folks who struggle with PTSD. But I also think it's interesting because if you believe these things sort of exist on a continuum, there's people with full-blown PTSD who've experienced an extraordinary trauma in, say, a wartime setting. And then there's also people, almost all of us, who experience at least some stress and trauma in our daily lives because, that's part of life. And presumably, many of the things that we can learn in sort of the extreme settings will read on how we think about folks in a more sort of ordinary setting. Because again, everybody in their life will have traumas. People you care about will die. You will have your challenges, et cetera. And so I think it's such an interesting and rich area. So maybe with that in mind, let's talk a little bit about specifically some of the work on PTSD and some of the things that you found, whether it's what's happening with some of the RNA-seq work that you did and discovering glucocorticoid signaling pathways and their importance for PTSD. If you want to sort of describe that journey for us.
Yeah. So PTSD is, when you first is described to you, you think it's like, oh, it's a tractable disorder, right? So you say, okay, there is a particular traumatic event. So if you place it in a research study, you like say, okay, the sample that I'm analyzing, was it before that event or after the event? So you say, okay, if it's before, I can do predictions. If it's after, is it in the immediate aftermath where the majority of the people respond, or is it like later where the symptoms are established? So you kind of like envision a lot of type of biomarkers you can. But then you realize a very critical factor is that there is individual variation, like you said, that those traumatic events in reality are more frequent than the disease is. So all of the people who respond, including the individuals that eventually will have symptoms, but they have something uniqueness of this maladaptive version of the response they're experiencing to that particular event that you need to kind of like parse out. So you need to naturally study vulnerability and resilience. You have to have populations that were both exposed to the same severity event. Of course, it's very difficult to know that because assessing trauma is based on the organism that experience it and not based on external criteria. But you could potentially measure the early signs of exposure that would be more. There will be maybe less variation. Everybody would somehow increase their cortisol levels and stuff like that. And maybe to a very small degree that you will see kind of like, oh, maybe the response of the PTSD subjects was not as high as you would want to, but in such a small percent change that, this single molecule is not going to help you. Like the original studies would go like, oh, can cortisol tell us? Because,
Right.
Measure cortisol, probably from that class, you realize maybe they said, circadian rhythms counts. When did you get the sample? Did they eat? Did they do that? Like, all those details. And the effect is so small with all those co-founders, it didn't work out. Right? It's not a single biomarker disease.
Right.
But, you envision that, and then you imagine that some transcriptional change happen that produce some protein changes that then these are lingering on. These are the profiles that you could potentially measure.
You mentioned, by the way, not a single biomarker disease. It's interesting because the very first paper in sort of this work that you'd sent to me, which was this 2014 PNAS paper on expression profiling with glucocorticoid receptor signaling. Basically, to sort of summarize it, looking at rodent models of PTSD, it appeared some rodents were more or less vulnerable to PTSD. Glucocorticoid receptor signaling was basically a biomarker of vulnerability. Yeah. And the part that I found most interesting in that paper, and I'm curious sort of the follow-up on this, was that preventative corticosteroid treatment, which is basically their version of cortisone, right, reversed the PTSD phenotypes. Now, I realize this is in rats, and we're measuring the reaction by do they run through mazes, and the stress that we're doing is not, watching an IED in Iraq. It's probably not enough food or something. Anyways, I'm curious, has there been downstream work like this with humans? Are there treatments for PTSD that people are at least contemplating that are related to the signaling pathway?
I think, yeah, exactly what you said. The right term is the downstream, right?
Yeah.
So it might be that as a master regulator, indeed, comparing all possible master regulators, I saw that most variance was explained by GR across tissues. And then I was like, okay, GR, I know that it will affect thousands of genes, but maybe if I boost all the population of the rats to a high degree so that there is no animal that doesn't have enough cortisol when they need it, which is in the immediate aftermath of the traumatic exposure, they need to have high cortisol levels. So no matter if they had some variability, some of them could just produce it, some of them not so much. I was making sure that everybody's boosted to what they need to have was good enough that on a population level, on average, they seem to be protected from what they would have happened to them after the stressor, but actually to a 30% of them would have happened, right?
Right.
But I think the key, as I said, is the downstream thing. That is basically a combination of profiles that is much more nuanced and tissue-specific, cell-type-specific, that could be the biomarker. Now, also the treatment, right? When you give cortisol, kind of like will affect all the cell types in the body. Not a great treatment. We know that it's a miraculous drug and saves from a premature newborn to a lot of conditions, but we want it to be something that, will be partnering with something else to make sure that it goes to the cell type and affect GR signaling there, potentially not with a crude all-in agonist, but maybe a more nuanced agonist that kind of like will require some co-activators and, stuff like that.
I love this, the not to go too far down a rabbit hole here, but I think that the talk about the nuanced agonist is really interesting because one of the things, like I'm not a doc, I'm not an MD or a PhD. My wife's both. And so I know how much I don't know. But one of the things is somebody who's not a scientist, you think about these things like, oh, cortisol, it's really good for these things. Why don't we just take it? It's sort of like your immune system, it's terrific at killing pathogens. Why don't we just dial it up? And you don't think about the fact, well, if you dial up your immune system too much, we call that autoimmunity. And it is like there's all sorts of bad things that happen when with inflammation of your immune system being dialed up too much. And similarly, most of these things that are very good for us for certain things are harmful in other ways because they are driving cellular activity that maybe if you're a premature newborn, you absolutely need to survive. And so it's okay that there's these side effects, but in a normal context, it's not okay. And so what it means is we have to be subtle, or I guess we would say like personalized medicine, right? Yeah. As far as this is concerned. And so I guess in some ways where I'm guessing this leads is as we think about PTSD and how we treat it, understanding what's happening if I'm the person who's struggling with it, what's happening to me cellularly is maybe different than somebody else. And as such, cortisol is a good treatment for me or not. If I don't actually need more cortisol, giving me more cortisol is harmful and it is not only unnecessary, but probably harmful.
Yeah, and also there are a lot of other very potentially supported treatments that people have actually tried by just taking theoretically and making hypotheses. For example, CRH receptor antagonists. They fail too. Why? Because there is not this nuance to who this is a good treatment. And we have this conundrum like PTSD and depression, so comorbid. Many people have both diagnoses, like both sets of symptoms. Nevertheless, how the GR signaling and CRH dependent signaling is somehow different in such two overlapping topics that, it needs a lot of like a nuanced understanding and stratification to really nail it. We know all the players, we might have some good agonists and antagonists for those systems, but nevertheless, we haven't done really the system biology version of clinical trials to really match people with the right drug.
But I want to touch on something else here. You mentioned, you started to talk about the difference between PTSD and, say, major depressive disorder. And I know one of your more recent papers was on specifically doing some RNA-seq work on the difference between PTSD and MDD and the different sort of transcriptional signatures of these cell types. Talk a little bit about what you found there, because I thought that was pretty interesting.
Yeah, so they do like a post-mortem studies that we do, and we do it in multiple ways. The major new transcriptomic technique is this single nucleus RNA sequencing. And we basically get samples of PTSD. So basically these humans died while they had the diagnosis, and they could have variation on how they died. Some of them could have died with suicide. Most of all of them, some degree of depression symptoms they had. So it's even a more difficult question. Basically, we are discussing analyzing PTSD with depression against depression only. So the depression only group is a necessary group when you study PTSD subjects' brains, and then the controls. So you need to have cohorts of postmortem brain cohorts where trauma was measured in enough degree to be reliable, right? So, and that trauma could be early life, later in life. It could be repetitive, could be a single documented. So we start doing those studies, and then we wanted to invest big within our research academic funding, right? Put attention to, single-cell transcriptomics might push us to the other side. Because the first bulk RNA-only studies, not multi-omic RNA studies, did not have such an impressive signal. And then we said, okay, we will bet this, we'll have smaller sample size, but with this technique, would we have more power? And indeed, we saw that we had. By disentangling the cell type and measure single cell level of expression, we were able to see in our first study that at the RNA level of neurons, most signal lie for both PTSD and depression. And we are very into pathway analysis and also in relation to genetics, but in a deep way. So I was telling to the lab members, not any generic pathway analysis would tell us to really we found the pathways involved. We have to create a model system where we actually create the pathway we understand. So let's say glucocorticoid signal in neurons of humans, not a generic cancer cell line.
Right.
In their own cell type.
Your own bespoke cell line for exactly what you need.
Yeah, so then we say, okay, let's create the most expensive pathway we can think about, pay the money, create that, and say, does this controlled pathway explain variation that we see in the brains? Then I will believe it. And indeed, we did it, and we saw, okay, in the generic pathway analysis, PTSD and MDD, indistinguishable. Creating this pathway, even very controlled, with controlled dose, maturity, you name it, translocation of the receptor to the nucleus, everything. Then we saw a separation with in PTSD with depression, a positive correlation and a negative correlation in depression. So we say, okay, we believe that, and then this motivated NIH to fund us, and then we did the big study in where we did multi-omics of many more brains. We went from 30 to 50 brains to 300 brains. And then we saw, okay, we were right, but now that we can see the full picture, non-neuronal cell types are part of the picture, and potentially we are talking about the communication of neurons with other cell types. Mm-hmm. So this external guidance stuff we start seeing, we're like, oh, this is very neuronal. Maybe this is a circuit disorder in the end. Easy to tackle. No. It turned out to be that those external guidance molecules were more about the communication of neurons with the immune cell types. And it's more complicated. We realized that probably the layers of the cortex were that they are more neuron-specific versus more other cell types are part of the story. And this can explain the correlations that I did 10 years ago. We kind of think that the upper layer of the cortex has those cell types that correlate with the blood, versus when you go deeper, you have more the neuronal specific signals that I didn't detect back then because I was bulk signal and bulk signal.
Well, yes. So two things I want to pull on here. One is, I think that what you said earlier, which is it's more complicated, is generally the answer for all things in biology. And specifically for anything involving either the CNS, the central nervous system, or anything involving the immune system. It's more complicated is the default answer. The other thing, I love that the work that you're doing, especially when it contrasted with the work from 2014, because the move from bulk to single cell is one of these things that, like, for people who aren't like in the industry, I don't think it's something that they appreciate. But like a lot of what was happening with bulk sequencing is like looking for very coarse signals that are so strong that even when somebody described it as like you take a fruit salad and you mix it up into a smoothie, and it's like, even when you're looking at all the different cell types, the signal is so strong that you can still see that signal. And certain things were discovered that way, and it was a useful technique, but only so much. Yeah. Now with single cell work, we can actually look at what's happening on the protein level, at the RNA level, right? Like the methylomics, like the whole thing, multi-omic, cell by cell by cell by cell. And this reveals, so it's like, I always think of it as like you took a microscope and now I just sharpened the focus. Oh, look at all these things I can see now that I could not see before. And what's fascinating, of course, is I think it would be useless without ML. because at the end of the day, the data would be so overwhelming that no human could pull it apart. But now you combine the power of single cell and the power of ML, and all of these new discoveries now are accessible, I think, for researchers. It's a fascinating time.
Yeah, and also like the idea that now we can use LLMs to even define cell types. It's so creative. I think once I was told by a professor that I have too much lateral thinking for my own benefit. And I think now is the opposite. I think students require to have it, because if you cannot think in 5 different ways at the same time, you're not going to solve it. Like
I think
you have to do it.
Right. I think that's a very astute sort of general observation about what's happening with science. So I used to think about science is about you have to know the thing all the way down.
Yeah.
But now the tools will help the tools know the things all the way down, and our job as humans is sort of to connect to
make a lot of prompts, right? We have to make a lot of prompts to really get the answers we want.
Hypothesis generation is the original prompt engineering, I guess. So speaking of which, we're going to bump up against time here. But I love this idea that one of your advisors told you were sort of too creative, because the sort of last question I had for you is, there's only 24 hours in a day, busy physician scientist who also has kids, et cetera. There's only so many things that they could work on. I'm curious, if there was something, if you had more time, is there a problem you would love to be spending some time looking at because you think it's really fascinating, but you simply can't because the lab is already busy and you're already busy? And what would that problem be?
This topic, the topic of epigenetic inheritance that started with when I was doing these generational studies in the rats. But then, when I came to the US, although I had the opportunity to work on the Holocaust survivor samples and of their offspring, nevertheless, I realized that's a very hard topic. I cannot answer you if Lamarck or not was right. I don't have the answer, right? Yeah. But if I could have, endless funding that is not restricted to proving that I can solve disease, I think it's such a fascinating, fundamental human question that we need to know the answer. But I cannot tell you if this is going to solve diseases necessarily, right? Because when I was analyzing the samples, I was like, the sample size is ridiculously small. And it is small because, until you convince the systems to do those studies, eventually you end up with their own studies. It's not an experiment, right? So guess what? Individuals that have endured generational trauma are also the parents of their offspring. So is it a transmission that is pure epigenetic inheritance, or is it happening because traumatized individuals cohabitate because they have to,
Right.
And
The confounders in experiments like this are incredible, and even what is the experimental design in order to demonstrate it is hard. I understand why
in animals it's also very hard, right? Because you have like one mouse and then you kind of breed it, and you have the problem of the pyramid, right? That everybody came from only one initial pair breed in that colony, and it like totally confounded also in the mouse studies, right? And so it's kind of like a hard, but I was always fascinated because, my favorite topic of everything is history. And if I can link biology to history, like, can you explain why French Revolution happened? Like, was it like the famine of the previous generation that pushed everybody's biology into kind of inventing their brains French Revolution? No idea, but it fascinates me that
it fascinates me too. I think it's interesting about it is because it's hard to not think about evolution as something that happens on these massive time scales to the point where like, it's sort of not, it's millions and millions of years. I was always surprised when I, first read sort of Stephen Jay Gould stuff on punctuated equilibrium and like, oh, actually evolution, even DNA-based evolution can happen much faster than you realize, and not just for simple stuff like the moths are now black, not white, because the trees are now covered in tar or whatever. But like it can happen really fast, but epigenetic stuff happens in one generation. Anyways, I agree, but I also understand why like a young, like early career researcher wouldn't pick that as their topic, because your chances of having success in your first 5, 10, 15 years are pretty low. Which is not what anybody wants when they start their career. So fascinating. Well, Nikos, thank you so much for the conversation today. Such interesting stuff.
I really enjoyed our discussion.
Terrific. Well, we'll be in touch soon, and thanks again for being on the podcast.